Okay, let's just dive right into this. Welcome to the Explainer. Today, we're taking a seriously spectacular journey. Imagine plunging right through the surface of human biology into this hyper-realistic, neon-lit intracellular space. We're going to explore the microscopic mechanics of human immunity, specifically something absolutely crucial, the major histocompatibility complex, or MHC. If you've ever wondered how your body distinguishes between friend and foe on a molecular level, well, you are in for a visually breathtaking ride. So picture yourself shrinking down to the size of a single molecule. As we pass through the fluid-lipid bilayer of the cell membrane, just look at this vibrant cellular universe opening up around us. You've got these floating ATP particles everywhere emitting this really soft, beautiful luminescence, powering the whole grand scientific machinery of life. This right here is the stage for our cinematic microscopic dive into the body's ultimate defense mechanisms.
Section 1. The body's defense system. A microscopic dive.
In this vast, glowing battleground, we've got a core division in our immune response, right? The innate and the adaptive systems. Now, you can think of the innate system as your rapid, first responder local police. It recognizes the basic microbial components. But, and this is key, it is absolutely necessary to pass the baton and instruct the highly specific SWAT team-like adaptive immune system. And it pulls this off using a very special class of cells.
Let's meet the sentinels of this system. Antigen-presenting cells, or APCs. These include macrophages patrolling your connective tissue, dendritic cells scouting the mucous membranes, and B cells hanging out in the lymph nodes. When they encounter foreign invaders, they actually swallow them whole, a process you know as endocytosis. But they don't just destroy them and move on. No, they use them for intelligence gathering.
Section 2. Meet the MHC molecules, the master communicators. Once an APC has literally chopped an invader into tiny peptides deep inside its endoplasmic reticulum, it uses the major histocompatibility complex, the MHC, to alert the rest of the body. Notice how most of the MHC is encoded on a 4 megabase region on the short arm of chromosome 6. You can actually see the class 1, 2, and 3 regions mapped out right here. This region is incredibly diverse, creating over 1,100 common human leukocyte antigen, or HLA, polymorphisms in the population. That is a massive genetic library. But look at the call-out at the bottom there, beta-2 microglobulin. This crucial part of the MHC class 1 molecule isn't even on chromosome 6 at all. It's encoded by a relatively invariant allele way over on chromosome 15. The precision of this genetic coordination across chromosomes is just staggering.
Okay, so the crucial takeaway here is that there are two main types of these communicators presenting antigens to T cells, MHC class 1 and MHC class 2, and they have very different jobs. Class 1 deals with endogenous peptides, meaning things found inside the cell, like viral fragments or tumor antigens, and it shows them exclusively to CD8+ cytotoxic T cells. Class 2, on the other hand, presents exogenous peptides, things swallowed from the outside like wandering bacteria, and it shows those to CD4+ helper T cells.
Section 3. MHC class 1, CD8+ activation.
Zooming into this photorealistic 3D rendering of the MHC class 1 structure, check out the heavy alpha chain. It's composed of alpha 1, 2, and 3 domains, which are bound non-covalently to that beta-2 microglobulin we just talked about. This stunning structure really has one primary purpose, signaling the body's assassins, the CD8+ cytotoxic T cells. Because it's dealing with intracellular threats like viruses, it presents these tiny viral fragments. We're talking only 8 to 13 amino acids long, right on its surface. Now look closely at the top of the molecule, right there between the alpha 1 and alpha 2 domains. That's the peptide binding site, and it's formed by 2 alpha helical walls and a beta-pleated sheet floor. It acts like a closed groove. Actually, scratch that, it's almost like a hot dog bun pinched shut at both ends. The endogenous viral peptide is held incredibly firmly in place at specific points called anchor residues. The hypervariable genetic regions of your specific DNA actually dictate the exact shape of this groove, which determines exactly which viral fragments your cells can hold on to and present.
Moving on to Section 4. MHC class 2, CD4+ activation. Shifting our focus a bit, in this glowing microscopic landscape, the MHC class 2 structure looks, well, a little different. It's an alpha/beta heterodimer, with both chains encoded completely on chromosome 6.
This specific complex is recognized exclusively by CD4+ helper T cells reacting to exogenous threats, like swallowed bacteria that the antigen-presenting cell has actively hunted down and broken apart in its lysosomes.
Pay attention to the groove itself here. Unlike the closed-off structure of class 1, the binding site here is an open-ended, promiscuous peptide-binding groove. That means it can accommodate much larger fragments of invaders, typically 13 to 25 amino acids long. It literally dangles the evidence out the sides for the rest of the immune system to see.
Section 5. The immunological synapse.
So, watch what happens as a naive T cell approaches the antigen-presenting cell. The tension here is palpable. Boom, they connect. This stunning visualization shows what we call the immunological synapse. The T cell receptor accurately docks right onto the MHC peptide complex, and you can see these glowing, energetic signal pathways firing up between the two cells. The CD4 or CD8 co-receptors are locking in, essentially double-checking the MHC class. This physical, energetic connection is the exact spark that ignites adaptive immunity.
But here is a brilliantly elegant failsafe built into human biology, the mandatory dual signal requirement. T cells are incredibly powerful, so the body absolutely doesn't let them activate on just one signal. Signal 1 is that specific contact between the TCR and the MHC peptide complex we just saw. But that's not enough on its own. You absolutely need signal 2, the engagement of co-stimulatory molecules. For example, CD28 on the T cell must lock into CD80 or CD86 on the antigen-presenting cell. If a T cell gets signal 1 without signal 2, well, it becomes anergic, meaning it permanently shuts down or undergoes apoptosis. It's like turning 2 keys to launch a missile. It ensures your immune system only attacks true, verified threats.
Section 6. When immune tolerance fails. As we zoom back out from these hyper-detailed molecular dynamics to the macro reality of human health, we have to face a pretty harsh truth. What happens when this delicate balance fails? If T cells fail to recognize a peptide as self and fully activate anyway, the result is devastating autoimmune disease, where your body's assassins turn on your own tissues. And on the flip side, these exact same MHC genes dictate tissue compatibility, which is what triggers immune-mediated transplant rejections. Our very survival, day in and day out, hinges entirely on this breathtaking microscopic dance of recognition. Really makes you wonder, how well do you actually know the universe inside you? Thanks so much for joining me on The Explainer.